Chidambar again to our class, and we just, I think, finished almost at the right condition to give it to next to you. So now I request Chidambar to please share what you want to show about the series spectra of macromolecules.
I have 10 minutes or?
Yeah. We have 20 minutes left in the class, so take your time, please.
Okay, that's great. Perfect. It says host has disabled screen sharing. So
sorry. Yes, now it should be available.
Okay. Yeah, I can.
Yes, we can share.
Is the screen visible?
Yes, yes, visible.
Okay. So don't get scared, students, by the 47 slides. I'm not going to run through all of them. Okay. Hello, everyone. Hi. Good to see you again for the second time in a day. So the idea behind this is what I'm trying to do is, what Professor Arnab Dutta has nicely set up, what is the circular dichroism spectrum, how is it used in, or what are the basic tenets of this spectroscopy. So what I will do is I'll come in and try and show you how this is actually used in looking at macromolecules, which is one of the applications. There are many kinds of applications. That is, you can use them in looking at biological systems like peptides or proteins. But I'm primarily going to show you about how to look at them in synthetic systems. Okay, so I'll skip all this sort of boring detail. So I'll just try and pick things from this presentation, show you wherever it is important. So I'll skip a lot of things, so don't worry about what I'm going to skip. So I'm sure we're all very much aware that many things in life are chiral, starting from things which are, let's say, macroscopic stuff like galaxies down to even the atomic and the molecular, and some people say even some of the subatomic particles are chiral. So chirality is something which is ubiquitous to life, and it has a very significant part in the life itself. So these are some of the objects or length scales at which chirality is expressed. And something which I'm sure you all will relate to is this double helical structure of DNA, which is chiral. And if you actually look at it, it is indeed a macromolecule, or it's a polymer which is held together by non-covalent bonds. And it is chiral. It's a right-handed double helical conformation. And this was discovered way back in 1954. And these were actually the heyday times for macromolecular chemistry, when lots of activity was going on all over the world. And at around the same time, back in Chennai, Professor G.N. Ramachandran was trying to figure out the triple helical or the structure of collagen, which ultimately turned out to be a triple helix as shown in this cartoon. And so there was another Italian scientist called as Giulio Natta, who was trying to look at synthetic polymers. So you can see that the first two examples were biopolymers. And the third one is a synthetic polymer, which is called as polypropylene, which we almost all of us use in our day-to-day life. And in this elegant JACS paper by Giulio Natta, he found out that the polypropylene also adopts a helical structure as shown here in this picture on the right-hand side. Okay. So if you are more interested, actually you can go through this very nice article on the origin of stereoregular polymers, but I'll not get into this. So if you look at polypropylenes, which we come across in almost in day-to-day life, I just want to impress upon you that how chirality actually plays a very important role even in material properties. So if you take a chain of polypropylene, and then what you see is that if all the methyl groups are on the same side, that is either above the plane or below the plane, then you get a very regular backbone arrangement of the polymers. And once you have a regular arrangement of the polymer backbone, they can actually come very nicely close to each other. As a result, you get a very highly crystalline polymer with a very high melting point of about 180 degrees. And that is what we actually make use in our labs for various day-to-day items. And another possible conformation is that you can make every alternating methyl groups actually go back and forth with respect to the plane of the board or plane of the methyl chains. And that gives rise to what is called as a syndiotactic in the polymer chemistry terminology, which is has a slightly lower melting point because the arrangement is a little more disordered now. And finally, if you actually come to atactic, that means there is no correlation in the arrangement of the chiral centers along the polymer backbone. You get a completely atactic or an amorphous polymer. And this is actually at room temperature. The melting point of this is below room temperature. So I hope this example shows you that the by just having arranging the methyl groups along the polymer backbone either in a regular fashion or an irregular fashion, you can vary the material properties by enormous amount. That is going from a melting point below room temperature to about almost 180 degrees and beyond. So here again, the chirality of this polypropylene is what is the relative chirality between the two stereocenters is what is playing a major role. All right, so then what I'm going to do is I'm going to jump ahead and show you a few examples. So this is actually an overview of how chirality has evolved over the years. That is way back in 1950s, the example which I showed you is of chiral polypropylenes. That is when the whole field started of looking at chirality in polymers with the work of Natta and other co-workers. And that slowly evolved into stable macromolecules in the 70s and 80s. And in the 80s, there was this idea of chiral amplification, which has come about. But in this, I will just try and show you about one or two aspects which are currently being sort of used in research or currently being explored. And what does this have to got to do with applications, or what is it of relevance to you? So there are, I don't know if you guys are aware of polymers, but just to be on the same page, polymers are nothing but long chains of molecules which have the same repeat units along their backbones. Okay. And this is what we call it as a covalent polymer. But if you have instead of the monomers being held by covalent bonds, if the two monomers are now held by non-covalent bonds, such as hydrogen bond or let's say pi stacking or any other kind of non-covalent interaction, then that gives rise to this kind of assemblies which are called as supramolecular polymers. And people typically what they have done is people used circular dichroism, which we are all learning about, or the cotton effect, to look at what is the conformation of the polymer chains. So if I take, let's say on this side, this chiral monomer, although the molecule is chiral actually, it is the absorbing part does not have any chirality in it. As a result, it gives a flat line. That means there is no chiral conformation in the solution. Or if you want to look at covalent polymer, the polymer backbone does not have any chiral conformation, or it does not have any preferred chiral conformation. So it does not preferentially absorb either the right or the left circularly polarized light. However, the moment you go to, let's say, one of this chiral polymer, that is either a supramolecular polymer or a helical polymer of a covalent polymer, then you have one of the preferred handedness, that is either the right hand or the left-handed conformation. And that actually absorbs the incoming circularly polarized light, giving rise to this blue trace, which you see here, which is an indication of the chiral assembly. Is everyone with me on this, or?
Sir, why is the monomer being chiral and still it is giving a blank peak?
Yes, good question. So what we are actually looking at is, so the answer to your question will become clear if I show you one structure, okay? So assume this is the monomer, okay? And the the chirality is in this alkane part. Do you see this, Ghazal?
Yes, sir.
And whereas the absorbing part is the central part, which is the chromophore.
Okay, sir.
Okay, so that is why the central part does not remain chiral as long as the central part aggregates or stacks into a helical conformation.
Okay, sir. Got it.
Or if at all, let us say if I had a ketone here, which was in a chiral environment, then that would have given a CD signal. Does that help you, Ghazal? Or
Yes, about it.
okay, great. Somebody else has a question. So about what I told till now.
So, sir, please explain again, sir. This is
Okay, I will come to this in a minute. I'll explain you. So this is what is called as an induced circular dichroism. I'll come Rajesh in a minute. Okay. So this is what people typically have used circular dichroism spectra for. And but recently what people have really got excited is that not only you can actually understand to study the conformation of macromolecules, like if you're studying biophysical chemistry or biochemistry, I'm sure you would have learned that the circular dichroism spectra is used to study how much of alpha helix or beta sheet or other conformation of protein is there. So in addition to studying the conformations, chirality can also actually give rise to some of the functions which are shown on this slide. I will not go into any of the details of this. So that is why people are more interested in now trying to look at chiral systems from an application point of view. So with this in mind, I'll show you just two examples, one in dilute solution, one in thin films, to show you how CD spectra comes into picture. So in this case, what people have done is they have made this particular molecule, which is a monomer, which has a central part, which is let's say a di part or a pi conjugated system, which can absorb light. And on either side, you have a alkyl tail, which has a chiral center onto it. Okay, the S stereo center, which you can see. And if you take this molecule in a chloroform at about, let's say 50 micromolar, the individual molecules are actually very well solvated, so they do not aggregate. However, if you actually take this molecule in an apolar solvent like hexane or heptane, then actually these molecules are not completely solvated because the pi conjugate systems now wants to stack between themselves. And as a result, you see this kind of helical arrangement, which is shown here, comes into picture. And because of this helical arrangement, what you indeed see is the circular dichroism spectra. And this spectra is what is called as a induced circular dichroism. So what I mean by that is, although the individual molecule is chiral, if it record a CD spectrum of this in a solvent, in a good solvent like chloroform, it does not show you a spectrum because the chiral part is actually not absorbing. And only on aggregation or only on assembly, like as shown here through pi-pi stacking or through van der Waals interactions, the chirality which is there in the side chain gets actually induced onto the core, which is this pi conjugated system. Thus, you get a circular dichroism signal, which you can see here. So the black trace is at high temperature. That is, all the molecules are very well solvated. And as I slowly start cooling down the solution, the CD spectrum starts increasing, which suggests that I'm forming these kind of aggregates. So this is another application of circular dichroism spectrum, where you can actually study how non-covalent interactions are taking place and giving rise to a polymer. Does this help Rajesh and others, or?
Yes, sir. Yes, sir.
So you see the distinction between what Professor Arnab Datta was saying about intrinsic CD and about induced circular dichroism? Rajesh?
Yes, sir.
So do you see the distinction between induced circular dichroism and a normal inherent or intrinsic circular dichroism?
No, sir.
You don't see, okay. Let us say one thing. Okay, I'll go back to the. Okay, so can you see this last picture on the bottom side? I have called this a helicene. Can you see this, Rajesh?
Yes, sir. Yes, sir. Helical division.
Helical molecule, right?
Yes.
So here the molecule by itself adopts a helical conformation. And if you take a spectrum of this, you will get a CD spectrum.
Yes, sir.
Correct? You expect that, right?
Yes, sir.
That is because the absorbing part is itself chiral. So this is basically nothing but 3 or 4 benzenes which are fused in a helical fashion.
Yes, sir.
Okay. So
Okay.
here the part which absorbs the spectrum in the visible region are these benzene units. And these benzene units are actually arranged in a helical fashion as shown here. Okay.
Yes, sir. Yes, sir.
So as a